and gentlemen,
Professor Jack Willeford once said, "Even in this high-tech age, the low-tech plant continues to be the key to nutrition and health."
indeed. Hello, my name is Larson Mapaco,
And my name is Sudesh Kalu,
juniors from Relief International School,
Maputo, Mozambique.
and we are here today to present a project entitled The Effects of Early Probiotic Supplementation on the Germination of Arabidopsis thaliana.
The pressure to produce enough food intensifies as the world's population increases over time. The exponentially increasing global population is projected to surpass the scale of 9.8 billion people by 2050.
Yes.
With more people comes the need for more food. And since agricultural commodities are the world's main source of nutrition, it is important that agricultural output keeps pace with the ongoing population. However, various environmental stressors, including the looming threat of climate change, have put agricultural industries under immense strain. Another environmental stressor is the impact of soil degradation on the agricultural industries. Farming an adequate amount of food has become more and more difficult with the slew of deleterious effects resulting from this soil degradation. These detrimental outcomes include the depletion of vital nutrients and the elimination of critical bacteria and fungi that synthesize essential organic material for the plant through the soil.
This imminent future disaster calls for a change in the way we cultivate our food.
As a result, this study was conducted to investigate how probiotic bacteria like Rhizobium leguminosarum or RL, Bacillus subtilis or BS, and Pseudomonas fluorescens or PF can impact on the growth of Arabidopsis thaliana when applied to the seeds.
With regards to the experiment,
we carried out proper laboratory precautions such as preparing the necessary conventional equipments, the seeds, and the bacteria.
yes, the PF, Escherichia coli, and BS were strict on nutrient agar plates, while RL was strict on Rhizobium X plates. We used a sterile plastic inoculation loop to pick up single colonies and spread them on their cultivation plates. After that, we incubated the cells at 30 degrees Celsius for BS and Escherichia coli and 25 degrees Celsius for PF and RL.
The 7 experimental groups and the control groups that make up the different treatment groups are depicted in Table 1. Groups 1 to 7 are composed of different quantities and combinations of the 3 bacteria. The non-negative controls are the water-only and the Escherichia coli groups.
For treatment groups containing more than one species of bacteria, we took equal measurements of each solutions to make a 500 microliter mixture.
On seed treatment, the Arabidopsis seeds were provided by Flynn Scientific and were thoroughly sterilized before experimentation.
Exactly. Regarding germination and growth, the square plates containing the seeds were placed on a rack platform at 45 degrees slant under a fluorescent light so that the seeds could fully germinate. Regarding the growth and germination, the plates containing the seeds were placed on a rack platform at 45 degrees slant under a fluorescent light so that the seeds could fully germinate. After a 2-week period, the number of seeds treated and the number of seeds that germinated was recorded. In total, we had 233 seeds treated, from which 135 germinated and 98 did not germinate.
Moving on,
our hypothesis was as follows. The greater the number of probiotic bacteria from multiple genera, the greater the growth of the root of the plant.
To prove this,
we measured and calculated the average root length of the plants on each of the treatment groups. The total root lengths among the treatment groups are depicted in this box and whisker plot. The X denotes average values and the dots outliners in the data sets. We observed that the average lengths of the experimental groups, except from the group BS and PF, were larger than those of either of the control groups.
Indeed.
We also counted the number of branches per plant on each group. The total number of root branches among the treatment groups are depicted in this box and whisker plot, where the X denotes average values and the lone dots outliners in the data set. We observed that the presence of bacteria led to a higher number of branch counts when compared to the two control groups. We also observed that groups containing RL had the highest branch counts. We also observe that the number of root branches increased as the number of bacteria also increased.
Based on our results, we rejected the hypothesis that a greater number of probiotic bacteria from multiple genera would lead to an increased promotion of root growth. On the contrary, each seed behaved differently in each situation.
Yes.
In image A, for instance, the plant from the water-only negative control group had one continuous root which did not branch off in any direction. In images B, C, and D, the plants were separated in a similar fashion with smaller roots and branches. And finally, on image E, the plant contained one main root which diverged into 7 smaller roots that branched off into different directions.
Exactly.
In conclusion, the results obtained from this experiment can contribute to the knowledge needed to develop a viable alternative to traditional fertilizers and to make agriculture more and more sustainable for the sake of both agriculture and global survival. As Al Magino said,
always do your best.
Whatever you plant now,
You'll harvest later.